Hello my dear students, I hope all of you are doing very well. You are learning, you are taking new skills, you are boosting your knowledge, and I hope you are dreaming for a very good future. So, we are actually learning this subject called Pharmaceutical Biotechnology. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute, Greater Noida. So, we are actually continue our learning of Unit 2 in Pharmaceutical Biotechnology. Today, I am going to tell you a very important topic, recombinant DNA technology. Sometimes we call it as a rdna technology. This technology created a disruption in the field of biotechnology. Today, whatever the products, whatever the vaccines that is biomolecules are observing, you will be very much happy to hear these things are possible because of the rdna. Lots of new kinds of our biological products are coming, FDA is approving through this technology. So, what is this and what are the things today we are going to learn? Let me tell you, I am going to start with the rdna. I will give you some basic introduction of the rdna, recombinant DNA technology. Then I am going to tell you discovery of the recombinant DNA technology, how it was discovered, what is the story. Then what is the goals of recombinant DNA technology and what is the procedure of making rdna, and also techniques what are actually used in the rdna technology. So, sometimes we actually write these things as a like this rdna. Okay. So, you should not be confused. If somewhere you may see the rdna or if somewhere you see recombinant DNA, both the things are same. Now, sir, what is this technology? Please tell us something about that. You see, my dear students, recombinant DNA technology, it is a technology. This is joining DNA molecules from two different species. Here we are actually joining two that is joining together DNA molecules from two different species. You have DNA molecules from two different organisms and you are joining them together. The recombined DNA molecule is inserted into a host organism to produce new genetic combination. See, whatever we have seen in our molecular cloning section, almost we are actually referring these things. Here we have told about vector, you may see in detail about the same molecular cloning technology. For example, vector, in my previous lecture you have seen what is vector, what are the types of vector, how you can choose a particular vector. Basically, for your need, whatever the need you will be having, based on your need you are going to choose a certain vector. You have seen various kinds of vector in my previous lecture where I have said mentioned you as plasmid, BSC, D, okay, BSC, YSC, human, human artificial chromosome. Okay. Sometimes we can use also vector first. So, this kinds of that is a vector you can use for our rdna technology. Then you will be needing some donor DNA, a certain DNA which DNA having some good properties, and that property will make a good combination and it will give you very good results. Why? Because one thing is very much clear to you all, if I take to here DNA, all of you are aware from DNA after transcription what we get? We get actually mRNA, mRNA, messenger ribonucleic acid. From that messenger ribonucleic acid, what we get after translation? Let me write, we actually getting protein, we get actually protein. Now, this is actually your gene of interest or your DNA interest. So, what we will do? Vector and donor DNA will be cleaved by using the restriction enzyme. In my previous lecture, I have told you what is restriction enzyme. Various big number of the restriction enzymes are available, near about 3600. Out of that 800 restriction enzymes we are actually using very rapidly. By using those restriction enzyme, you will be able to cut this vector. See, it is cut it, and now this area this DNA, donor DNA, you can insert in these places. After mixing, after because ligase enzyme will be there, it will help for this kinds of sticking. So, these things will be after DNA ligase actually added, it will seal like this. Then these things, entire this complex will be inserted into bacterial cell. So, this is the bacterial chromosome, this is the bacterial chromosome, and our these things, this entire complex we actually inserted into the bacteria. Now, what will happen? This recombinant DNA molecules will be replicate and cells divide, because this is a common feature of a cell. Cells are actually very rapidly dividing. Okay. So, when cell will be divided within cell, our this thing, this recombinant DNA molecules will be also replicate itself. It will also divide. Cells will be also divide. Now, all of you are aware it has some instruction called DNA. It has some instruction, nucleotides. So, that thing will be also transcribed and also it will be translated. So, obviously, when it will translate, we will be getting some results. Basically, up to here we call it as a clone. Okay. Now, if I tell more, DNA molecules are actually extracted from different sources and chemically joined together. For example, DNA comprise the this thing called enamel genes may be recombined with the DNA from bacterium. This is the fragment, this is the vector, and we have got the recombinant DNA. Okay. Whatever previous my this picture I have tried to what I have tried to tell you, that thing I have actually summarized in this. Now, what is the story, sir, behind this? What is the story behind this? Who discovered these things? Here is a very beautiful picture that I have collected from internet. The copyright is actually going to them. You see, all of you have studied the genetics or genetic engineering or gene etc., in your school days. All of you have got a good knowledge about this scientist called Gregor Johann Mendel, where he has told you about the heredity in 1866. You also have listened about the various experiments done by Oswald Orwe, where they have shown the evidence of DNA as the carrier of genetic information. Then in 1950, you got to know about scientist Arrowin's rules, base of pairing, where he have told how it is happening. Then a great scientist for whom I have huge respect, great scientist Rosalind Franklin. They have obtained the X-ray diffraction images of the DNA. See, Watson and Crick, they have actually got Nobel Prize in 1953 after 1 year. After 1 year, Watson and Crick, they got Nobel Prize because they have told that DNA are actually double helix, they are actually joining together in a helical structure. They have published the picture. Before 1 year, Rosalind Franklin, she actually identified through using the X-ray crystallography, X-ray diffraction, the images of the DNA. So, these things actually happening. This is the story of our entire genome or our entire genetics, genetic engineering and genomics. So, after that in 1961, in 1961, genetic code was actually cracked by Marshall. I think you have studied this thing in your school days. And after that, see, before that people are not aware that yes, gene is responsible for our daily activities, our protein that is a creation, transcription, translation is happening, due to that we get some protein, and protein is responsible for everything. People are not aware before that. Okay. After doing this kinds of this 3 or 4 activity, these and this, people are also this. People are very much got to aware, they got to know that how the things actually happening. Now, what next? After that, the great things was actually happened. In 1970, Stanley Cohen and Harvard Boyer, they have developed recombinant DNA technology. They have developed that thing in 1970. And after that, we have seen a lots of development was happened, like Sanger sequence in 1977. Then we before that we also have seen that synthesis of first synthetic gene by Khurana et al. in 1976. Okay, when in 1970 Cohen and Stanley Cohen and Herbert Boyer, they have developed the recombinant technology. Next year Khurana, scientist Khurana and his team, they have actually published their synthesis of first synthetic gene. Then sequence was actually happened, sequencing, very famous Sanger sequencing. By using the technology you can sequence a certain gene or genome. After that Kerry Mullis, they have actually demonstrated, convinced and demonstrated the PCR polymerase chain reaction, and then in 1984 Als developed the DNA fingerprinting. Today DNA fingerprinting is everything, right. Today if somebody do some crime, people very fast forensic scientists actually go for the DNA fingerprinting. And after that there are lots of activities was happened. Yes, there is some time after a long time in 2002 you can see GeneScript become the first company to commercialize the gene synthesis. See, the story was started here in 1976. It has taken long year to commercialize synthetic gene, for to bring some synthetic gene. And yes, meanwhile in that time I have not mentioned, in that time our human genome project was also completed, HGP. Our human genome project was also completed. Then if I tell you more, in 2008 GeneScript became the first world largest gene that is a synthesis supplier, and yes, our human genome project was completed. And after that, I am doing some mistake. After that, after in 2008 Gibson et al, they published their synthesis of first synthetic bacterial genome, and then GeneScript became the first world largest gene synthesis supplier. And then in 2012 again GeneScript joined synthetic East 2.2 project in their first synthetic eukaryotic genome. And then 2013 Crick's in 53 later to his son describes the double helix cells for 5.3 million. See, Crick told his son about that double helix structure of the DNA when in 1953. So, scientists are actually aware when they are doing research, they cannot publish their research because they cannot reach in a conclusion without evidence. My dear students, if you are learning the subjects from the bottom of your heart, if you are giving 100% to your studies, especially for research, you have to publish your research, but for that you need the evidence, you need the data, you need the experimental evidence. Crick, that is failed to tell about the double helix, but he mentioned his son. After few years later, he and his that is with Watson, they have published. Okay, now whatever the letter he told, he written his son, it was actually sold in that money. And after that, we have got a new technology. This is called, let me erase. After that, we have seen the development of CRISPR, CRISPR, PR. Yeah, we have seen CRISPR is the gene editing tool. Okay. This is the entire story of our entire DNA, gene, genome. Okay. So, you can see where our this technology is actually that is relying this Stanley's and Cohen's recombinant DNA technology. Now, I already mentioned about these things. Now, let me tell you something in a very great detail. So, when the discovery of DNA structure was done by Watson and Crick in 1953, then isolation of DNA ligase was happened in 1967. Then isolation of restriction enzyme and endonuclease was happened in 1970. Then Paul Berg generated the RNA technology in 1972, and after that Cohen and Boyer in 1973 produced the first plasmid vector capable of being replicated within the bacterial host. See, I am trying to focus on up to this area, whatever I have told you, I am zooming this here. Okay. From 1953 to 1973. This is the story how the things are actually progressed. Now, what is the goal of recombinant DNA technology? First goal is to isolate and characterize a gene Okay. Then to make desired alteration in one or more isolated gene, then to return altered gene to a living cell, then artificially synthesize new gene and alternating the genome to an organism, understanding the hereditary diseases if we can cure that thing, and ultimately improving human genome. These are the main goals of rdna technology. Now, very important question or very important interesting topic or interesting area we have raised. Procedure of making rdna technology. Sir, you have told us lots of things, its benefit, its story, its history, scientists who are actually working in this field. Now, tell us something about the procedure, how we can work with the rdna or how we can make rdna. So, for that first you have to isolate a certain DNA, basically the DNA or of interest, of gene of interest. Then you have to cut the DNA, then you have to join the DNA, and lastly you have to amplify the DNA. Isolation, isolating, cutting, joining, amplifying, isolating, cutting, joining, amplifying, ICJA. Always remember, many of the times students actually do not able to recall what are the steps: isolating, cutting, joining, amplifying. Do not worry, let me make the things much more easier. So, isolating, how you will isolate a certain DNA. First you need to do the DNA extraction. How you will extract the DNA? For example, this is a cell. Cells are actually lysed by using the detergent that disturb the plasma membrane. Then the cells contents are treated with the protease to destroy the protein, and that is a RNase enzyme to destroy the RNA. Then in next step what we will do, there will be cell debris will be plated in a centrifuge. The supernatant, those are liquid will be there containing the DNA, it will be transferred into the clean tube, and the DNA will participate in with the ethanol. It forms viscous strands that can be spooled in a rod glass. This is the first step of isolation of the DNA. Now, the next step is the cutting of the DNA. So, DNA can be cut into large fragment by mechanical shearing. If you do some mechanical that is a stress or shearing, it will be divided, you can cut. Then you can also use restriction enzyme, restriction endonuclease, what we are actually telling you in our previous lectures, how you can use restriction endonuclease enzyme to cut a particular DNA. So, this will be work as a scissor, it will cut the molecular genetics, this will be work as a scissor to cut a DNA. Then, yeah, here is the picture. So, this is the DNA complex, here is the DNA complex, and you can see the restriction enzyme, it is actually cutted, it is actually cutted the DNA, yeah, okay, up to here, yeah, here, yeah, it is actually cutted in these areas, you can see. Let me make it much more clear for you people. Just a second. Yeah, in this way. Okay, it is cutted. So, a special class of sequence, a specific enzyme, it is actually found in the bacteria. You are aware about, because I already have mentioned in a previous lecture. Site specific cleave of DNA molecule at a specific nucleotide sequence, and this restriction endonuclease enzymes recognize the DNA based sequence that are palindromic. That are palindromic means again and again it will be there. This that is restriction endonuclease enzymes make stacked cut with the complementary base sequence for easy circulation. Then what is the next stage? The next stage is the joining. You have the plasmid, and you have taken the DNA. See, you have taken the DNA, you have taken particular DNA area. So, this will be work as a foreign DNA. This is a cutted DNA. Whatever the DNA actually you have cutted, or DNA of interest, DNA of interest. Okay. So, what we will do? Let me tell you in a very nice way. So, this is the foreign DNA, these things will be cleaved. This is the your plasmid. So, your plasmid will be, it will be also cutted, and then both the things will be joined together. Okay, and helium will be there, and your DNA ligase will be joining these things, and this will be called as a plasmid chimera. Okay, joining. After joining, what is next is this is called amplifying the recombinant DNA. So, transforming the recombinant DNA into a bacterial host strain. The cells are treated with the calcium chloride, DNA will be added, the cells will be heated up to 42 degree centigrade, and DNA goes to a cell by somewhat unknown mechanism, and one in the cell the rdna will be replicated, and when the cell actually divides, the replicated recombinant molecules go to both daughter cell, which themselves give to the divide. Okay. So, this is the process or procedure by which you can make the recombinant DNA technology. Now, what are the enzymes or what are the that is proteins, enzyme proteins are actually using, or what is the reason, what is the their activity function. So, DNA ligase we actually using to bind the DNA molecule. You have seen in our previous area, to bind our DNA molecule. These things, both the things will be joining because of the DNA ligase. Type 2 restriction endonuclease cleave the DNA into the specific site, but it is basically cutting. Sometimes you actually use the reverse transcriptase, make DNA copy from RNA molecule. Yes, sometimes it will be required to make DNA copy for RNA molecule. Then DNA polymerase filling single strand gaps in DNA duplex. Then polynucleotide kinase add phosphate to 5-OH2 polynucleotide. Then terminal transferase add homopolymer to the 3 prime. Okay. Then endonuclease 2, it remove the nucleotide residues from the 3 ends, and bacteriophage, that is lambda endonuclease, it remove the nucleotide from 5 ends. Also you can also use alkaline phosphatase, it remove the terminal phosphatase. Okay. So, these are the various enzymes which are actually widely used. Now, plasmids vector actually widely we are actually maximum time actually using plasmid vectors. Here you have seen plasmid, let me remove the inks, plasmid, maximum time we are actually using plasmid. Now, let me tell you something about the plasmid. If you missed my previous lecture where I have told you in detail about the plasmids, this is a very small or circular DNA that are separated from the rest of the chromosome. They replicate independently in the bacterial chromosome and useful for cloning DNA insert less than 20 kilobyte, kilobase. Insert larger than 20 kilobytes, it will are lost easily in the bacterial cell. Okay. Now, few are the techniques we are actually using very rapidly in rdna technology. First is the gel electrophoresis, then cloning libraries, restriction enzyme mapping, PCR, nucleic acid hybridization, and DNA microarrays. I will tell you more in my upcoming lectures how and why we are actually using these technologies. For example, gel electrophoresis, you have protein, you have various kinds of protein, you can see their size is different. You want to separate this protein, you want to separate this protein. See, these things are falling one size, these things are falling in other size. So, you can separate protein by using the gel electrophoresis. Cloning library also you are aware, we also need the restriction enzyme mapping. So, these techniques are actually we widely use. I will tell you more about all these techniques in my upcoming lecture. That is why I am not explaining right now. These are things are very interesting, and you have to know in detail about all the various techniques, gel electrophoresis, cloning library, other things. So, at the end of our we are actually now at the end of our lecture. So, what are the things today we have learned? We have learned about the recombinant DNA technology, the discovery, at what stage it was actually discovered. We have seen the entire landscape from Gregor Johann Mendel's that is a hereditary concept to CRISPR, where our rdna technology was actually developed, what are the story behind that. We also have seen what are the goals, what are the procedures, what are the techniques are actually used. So, these things we have learned. In next lecture you do not miss, it will be very important and interesting lecture. In that lecture I am going to tell you about the genetic engineering. I will tell you very few facts how you can use genetic engineering in biopharmaceuticals, medical science, animals, vaccine production, DNA fingerprinting, also in insulin production and also PCR, and this leads to grow the plants in dark environment. Yes, in dark environment you can grow some plant without sunlight, can you imagine. So, a very interesting lecture is waiting for you in my upcoming session. So, thank you all of you Thank you my dear students for being with me for last 24 minutes. I wish you have learned a lot of things, and I wish you will implement all this knowledge, all the skills and all the wisdom from me whatever you have learned in your research, in your studies. So, thank you all of you, God bless, all the best, we may meet in our next lecture.